Hello, I'm Hal Weiser. I'm a soil health specialist.
Hello, I'm Susan Sampson-Liebig and I'm a soil quality specialist.
And I'm Mary Podall. I serve as a state conservationist for USDA and NRCS along with my counterparts here.
The first stop today will be at the Menoken farm where we're going to learn about soil health.
The second stop of the day will be the Agriculture Research Station in Mandan. We will be seeing the research that they've done long-term over 30 years for soil health.
So we have a lot to see today, don't we? Let's get to it.
Hi, I'm Darrell Oswald.
And I'm Jay Fear.
And we're at the Menoken farm today, which was established in 2009. The Menoken farm is a combination of natural resource education and systems approach conservation, where we employ the soil health principles and move towards soil regeneration.
And today we're going to look at high tunnel garden production, along with sunflower production with cover crops and corn production with cover crops as well.
So let's get to it.
Hi, we're here today with Jay Fear, conservationist with the Burley Soil Conservation District. And we're at the Menoken farm, and we're currently in the high tunnel. Jay, you want to explain what the process here is with the high tunnel?
Sure, Hal. I think the goal in the high tunnel is to create an ecosystem. And in that ecosystem, we want it to function. And so that ecosystem requires high plant diversity. It requires the pollinator world, requires water, and requires soil. And in a high tunnel, I feel it should always require cover crops. And so we start to connect all of these increments together. And we create this ecosystem then where we can go ahead and cycle nutrients over and over. I don't have to bring in outside nutrients, so everything you're seeing in here, there's no artificial fertilizer applied, there's no insecticide, and there's no pesticide, and there's no soil disturbance. These are all the components of the ecosystem that you want to create. So we want a more natural predator-prey relationship. So if we have scenarios with either leaf disease or insects, etc., we try to handle those in terms of the compatibility of the species. And it allows them, instead of finding just one type of species in a bed, you might find any of these raised beds or lower beds might have multiple species in them. So that's kind of the goal initially, Hal, is create this environment with the ecosystem.
And your system starts in the fall, right, Jay, with the cover crop?
I think that's why we're standing at this particular bed right here. This is a good example. So the harvest has taken place on this one. I think we had 7 harvests of lettuce out of here. And so now we're going to plant it to a cover crop mixture. And so if we plant it to a cover crop mixture, that's the opportunity for us to have some things happen. It's going to be sunlight harvest and CO2 harvest. And we're going to bring carbon into this soil. And we're going to rebuild the structure on the soil. And so what we'll do is we'll build it around cereal rye. And so I'll start out probably 50–60% of that mixture might be cereal rye. And then from there we'll add some additional species. Phacelia, depending on the crop rotation, a little bit of brassica. That starts that cool season. And then depending on the rotation, it might bring in a little bit of field pea. And so we start to build that mixture. Now it's North Dakota, so over winter everything's going to die out. What we're looking at then is we're only going to have cereal rye in the spring. That's the only thing that's going to survive here. And the cereal rye, there's no crop disturbance to plant the garden. And so when the cereal rye is about a foot tall, foot and a half tall, we'll smother it, take away the sunlight. So we'll put a canopy over it for 10 days. Take away the sunlight, cereal rye will go down, and then we'll plant into it with minimal disturbance.
So you just open up the residue and plant your seeds?
Actually, I take a scissors.
Scissors?
Yes, I take a scissors because the residue is going to be thick. I'll take a scissors and I'll cut through the residue. And then I'll make a small opening for the seed in that area that I cut. And then from there we'll take the drip lines and we'll lay the drip line right next to the seed trench. And so the drip line, sometimes you have to replace these after a year. It's got the micro pores, but they're very efficient. And the thing I really like about them is there's no soil splash. So if you take a look at the leaves in here, you don't see soil on them. And so if you get soil on your leaves, of course, now you're starting to ask for some potential disease issues as well.
So you talked about multiple species, and if we look at this particular box, what species are growing in here?
Yeah, so this one was sweet potatoes with sweet peas. And so we're looking at a scenario here where the sweet potatoes took over the sweet peas. The sweet peas were pretty light. You don't want to seed them so heavy. That's just your companion. And then the sweet potatoes took them over and smothered them down. But it gives you that crop diversity and gives you an opportunity to connect underground. The next one to me is the most interesting one.
Well, I see flax in there.
Yeah, yeah.
And carrots, obviously.
Yeah.
What else do we have in there?
Well, if you look a little closely, the carrots got very robust. But if you look closely, you're going to see the onion rows.
Onions.
And so the onions are developing nicely. And so there's 3 rows of carrots and 3 rows of onions in here. And the interesting thing about it then is we put a smattering of flax. And so the flax, as you know, is highly mycorrhizal fungi. And so now we've created a scenario that I think fosters life pretty well. A lot of gardening is who likes who. And the onions and carrots like each other. And so in that particular environment, we add a little bit of flax because then we think back, how did our soils evolve? Well, and our soils evolved, yeah, with a lot of plant diversity. And so here what we're doing is just, again, it's in the ecosystem of this high tunnel, but we are putting together a scenario that mimics that just a little bit. And then right next to it are the annual flowers. So again, you want to create something to attract the pollinator world. So we usually have the doors open, and so there's a lot of access here. They can come and go. And so it's a different world than shutting the door and trying to not allow something to come in. Okay? Here you're opening the door and you're welcoming the pollinator world, the insect world to come in. And so there's 7 different annual flowers in here. And I think color is such an important item too in the pollinator world. You know, yellows and reds, you know, they're a strong attractant. And then as we keep going down, we get to the cucumbers, and the cucumbers had an understory of flax planted with them. And so the cucumber flowering has been pretty profuse and intensive, and we've had a good harvest on the cucumbers, but it had an understory of flax as well. And of course, once the cucumbers got about half grown, they just took over the flax.
So one of the other things that you've added to this, most of it is annuals, but you do have some perennials. You have, well, some of these herbs are perennials, and then you also have some trees you've added.
Yes, there's 4 trees in here. Cherry tree, and then we have a peach tree over at the edge of the flowers, so the peach tree. And then we have an apple tree down by the tomatoes, and I think there's 2 cherries, a peach, and an apple. And so it really takes up no more room. And so it's very efficient in your ecosystem.
And applying the soil health principles.
Right.
You're providing that education. And,
it's the foundation of the ecosystem.
We're back with Jay at the worm compost bins. Jay, you mentioned that you use bioinoculants in your greenhouse, and you also use them out in the field, right?
Yes.
On the Menoken farm?
Yes. So we start with you know, a completed compost. And so typically we make them in a bioreactor, kind of like the round black structure right there. It has aeration tubes in it, cardboard tubes in it, and it keeps it aerobic. And we add the water to it and to the organic materials, and then over the next few months it turns into a static completed compost without turning, which enhances the fungal community. From there it goes into these bins and we add the worms. And sometimes we add the worms even after the thermophilic stage is done. After the heat stage is done, we can add the worms even into the bioreactors. But for sure, when they come into these worm bins right here. So the job of the worms is to consume this material, bring everything through their stomach.
So how long does this process take, or how long do you have the earthworms in the system?
Okay. So we started, the bin will be good for at least a year. And I think we can get a year and a half to maybe almost 2 years of worm juice coming, slowly seeping out of the other end. So the cardboard tubes are for aeration. They'll actually eat the tubes. So the tube will eventually just be this top part left, but the tube in the compost itself will be decomposed and eaten. And then there's a little bit of a slope on the tubs, and it brings it down and into a container. And from there you have worm juice. And we typically send it in for analysis once a year to the Molecular Research DNA Lab in Shallow Water, Texas. And that's been a real interesting process. It takes a better part of a month to complete it. And what they'll do is identify all the DNA that's in that liquid. The goal of the worms is to consume all of this material, and then as we add water, it slowly leaches everything to one end. And after we've done this for about 2 years, then whatever's remaining goes into the garden. But this is what we actually apply onto the seed on the cropland, onto naked seed, sunflower seed, corn, wheat, soybean, etc. And so we put on approximately 4 gallons to the acre in the cropland, right into the seed trench with the seed. And when we do the garden, the high tunnel garden, the outdoor garden, we inoculate the seed with the liquid. But we can take a look at them here, Hal. See what we find. So you can see we've got them in here. They're eating, this material's not fully decomposed yet. They really can't eat organic material, like something like this or even a big piece of straw, because they have no teeth. And so it needs to get decomposed to some extent. And so that's why I kind of like...
The microorganisms slowly decompose that before the earthworms can actually decompose it.
Yeah, and it's an intensive process, because then the microorganisms inside the earthworm, inside the red wiggler, is going to decompose again. And so you've got this recycling, if you will. And then you start to get, when you get the end product, the liquid, it's not a fertilizer. There's very low nutrient levels in it, but there's very high levels of soil food web in it.
So when you've done some of this analysis, or had this analysis run down in Texas, the DNA analysis, how many different species have they identified in some of your samples?
The last one we did was 350 species.
350?
Yeah, and it was roughly half fungi and half bacteria, but about 350, 2 years in a row.
Yeah, that really points out the benefit of the static compost. You're not going to get that in that high fungal level necessarily in...
Yeah, much more difficult to achieve when you turn the compost in a traditional manner. Whereas if it's static, it's much more fungi friendly. It's like not rototilling your garden or not cultivating your cropland field. So you make it a better ecosystem for it to survive in. You can purchase bioinoculants, Hal. You can go online and buy them and apply them. But to me, I think it's something we can make ourselves. So we can take a bioinoculant, and the germination tests we've done on this, it helps foster our germination levels, and it gets the plant off to a nice start. So I like that, because now the seed is germinating. If you're going to use any of this liquid, any of this worm juice, you want it on at germination time, before the seed germinates. And then it germinates in this high microbial environment. And that's a bit of a game changer. And so, you can, again, you can buy them, but I think you can also make them. And I think that's kind of the interesting part of it all, is you can take your materials that maybe normally would have ended up in a landfill or something like that. You can gather up as many of those materials as you can, and you can start the process yourself and make your own material.
So we're standing here on the edge of Field 3 here at the Menoken farm. Prior to what you're seeing here, this was a monoculture of wheat for 12 consecutive years. And the SCD Board of Supervisors wanted to see how a monoculture would fare with our cropping system, but after 12 years they had a change of heart, and so here we are. And so late last winter, we actually started to reintroduce some biology, some cover, some things on it, and we fed on this side. When it came time to seed this spring, we decided to go with a diverse cover of sunflowers, solid seeded sunflowers, and 7 broadleaf cover crops along with the sunflowers, and so that's what you see here. And as you can see, considering what we've endured as far as lack of moisture, we are seeing some results. You see the canola, the cowpea, the soybean, the phacelia, and what this adds is something that this field didn't have before. And so again, previously it was an annual of wheat for 12 years, which was a cool season grass. And so by bringing in all these different roots and root exudates, we're giving the field something it doesn't have. And so we're really blasting it with diversity. After harvest, if we're able to harvest, we'll have some biomass and some structure in the field, and we'll be able to add another one of the soil health principles of hopefully integrating livestock. And so we'll have biomass to eat and to feed our livestock. So we're excited about this field. It's something that employs quite a few of the soil health principles, and we're starting and hopefully seeing some success with it.
So one of the things when we're looking at soil health is to have a pretty simple tool with, and that would be a tile spade or a shovel, and dig in the ground and just look at the soil structure. Look for any horizontal platiness that may be in the field. Dig a few holes, get an idea for what the structure looks like. So this field was in continuous wheat for 13 years, as Daryl mentioned. Before that, it was conventionally tilled, often plowed, right? So we had structure that's remnant structure from that tillage. So what we're trying to do in soil health analysis and assessment is look at the soil structure, look for this platiness, if it exists. How strong is it? Does it break apart fairly easily? So I'll dig up a sunflower plant here. So as we dig that sunflower plant up, take a look at its root structure. And sometimes if you do have a pretty strong compaction layer, you'll have what we call a J-root, where the main taproot will come and it'll hit a restriction and it'll grow sideways. This particular plant doesn't. Has a fibrous root system and appears not to have any difficulty rooting in this particular environment. I'll go look at another one just because we typically look at more than one plant in a field or one area to get an idea of what's going on. And then you can also have wheel tracks that cause compaction. So you want to look down the rows, see if there's any compaction identified within a wheel row. Then you maybe want to look at a plant that's shorter, perhaps. Here's another sunflower plant, but it has a main taproot that goes straight down, doesn't really have a J-root on it. So No real indications that you have any rooting limitation with those two plants we looked at. Take a look at a canola plant. Now you see that canola root had a little difficulty rooting past this 1-inch layer, where had to take a little bit of a turn. So there was a little bit of root restriction, but it did find a crack and break through it. So not too bad. We have a turnip here, and you can see what happened to that turnip root. Kind of came down instead of having a main tap root going straight down. It did take a J-turn, as we call it. So it had a little bit of difficulty at about a 2-inch layer, 2-inch depth in the soil. What we want to do is get some of these tap-rooted plants in here to break up some of those compaction layers. So just by visually digging some of these up, you see where, we're doing that. Some of these plants are going straight down. Some have to take a little bit of a turn before they start going vertical into the soil. Again, we'll dig these plants up and see where they are at for compaction. They'll be our last two plants we dig up on the field. Here is a canola plant. It's got a taproot system. Let me get the soil aggregates off there. And you can see that one does have a little bit of a J root, kind of at 1-inch depth. Had a little trouble getting through. It doesn't have a main taproot going straight down vertically. So there are some compaction problems near surface on this field after being in wheat for 13 years straight. And then we'll dig up the sunflower. See if we see the same thing. Okay, again, we don't have a main taproot going vertically into the soil. It kind of goes off to the side a little bit. Probably broke off, but had to detour a little bit because it hit a compaction layer. Other than that, it has a nice fibrous root system, healthy plant, but it's having a little difficulty penetrating those compaction layers. But that's what these plants in a diverse rotation will do. Over time, you'll start breaking up that compaction layer. They might struggle a little bit first few years, but slowly you start breaking down that compaction layer. Just wanted to point out one of the other indicators we have, and that's surface residue. If you look at the surface of the soil here, it's 100% covered with residue, which in some cases it could be an impediment for getting a crop to germinate if you don't quite do the planting practice correctly. And there is a little bit of bare in here, bare area where the plants didn't come up, so there might have been some germination problems. But from a soil health perspective, keeping the soil covered, keeping the residue on the soil surface is something we really want. We want to protect the aggregates, the soil aggregates, from raindrop impact. Once you have a raindrop that hits the soil surface, the aggregates can bust apart, then you get surface sealing, then you get more runoff. A lot of things go along with that. So keeping the residue on the surface keeps the soil temperatures cooler, and when it's 100 degrees out and the sun's beating down, that's pretty important too.
We're standing here again in the middle or on the edge of the 60-inch cornfield, and this 60-inch cornfield does have some covers in between, and of course this is obviously we're adding more diversity with the covers, but we're also using sunlight, or trying to capture more sunlight, and to get some vigorous, well-growing corn plants. And so this is a step away from the traditional 30-inch corn that we see. Obviously the 60-inch spacing allows too for the covers to be planted in between, and this particular mix of covers that we have in here. We have some soybeans, some sorghum sudan, some German millet, some Italian ryegrass, some turnips to help again accomplish aggregation and have different sets of roots in the soil. The biomass that will be hopefully produced with this corn crop, if we're able to harvest the corn, will be excellent again for integrating livestock into the system. We see another addition of the soil health principles in the 60-inch corn.
Daryl, you mentioned this field's been in diversified crop rotation and now no-tilled for 14 years.
Yep, since 2009, yep. It's had all the soil health principles applied to it since then.
And we started out with a soil that did have a compaction layer, if you want to call it that horizontal platiness from many years of conventional farming, correct?
Conventional farmed probably up to 100 years prior to what we've seen here at the Menoken farm, and then in the early time up to the Menoken farm, the use of a Concord air seeder predominantly, which was very common for the area.
Mm-hmm. So if you dig a hole, Daryl, we'll see if the structure has improved in this particular field. Granted, it is dry, isn't it?
Yes, it is. Yeah, it's.
So if we're going to look at this soil, we can see we got good granular structure in that upper part. Breaks off very nicely into aggregates. And as we go down, if we take a look at this soil, you see the roots. You can see some earthworm channels in that particular soil. So what does that do? That adds some
obviously
porosity to that soil, right?
Porosity. You know, we're capturing, the goal obviously is to capture every raindrop, and that's especially important on a year like this year because it's been very limited as far as the rainfall. So
far you can see that granular structure is starting to take hold even in that old platy structure from the plow pan at even in that 3 to 5 inch depth. And even as you keep going down, you can see the roots are having no problem going through there. Yeah, there is an earthworm kind of curled up saying, hey, this is a little too dry for me. But finally we get down to this structure on the lower end of the which we're examining here. There's an earthworm doing its job, creating the soil pores and helping to break down that soil structure. So there's another good example. See all the earthworms kind of curled up saying, hey, it's way too dry, but give me some water and I'll keep doing my job.
one thing that comes to mind for me, Hal, when I look at this again and stuff is the color of the soil from the last field. Mm-hmm. And I think we're seeing obviously that this is a little darker or somewhat darker. And
yes, that's one of our indicators, Daryl, and Susan does have the soil Munsell color chart.
Hold that.
Yep. What we do is like to compare soil colors and the fields and the way they've been managed, and we did not color the other field, but we can look at this. So the best way to do soil color is put the sun at your back shoulder. And this is a little bit moist, it's not dry. So what we do is we put the soil underneath the Munsell soil color chart, and you can see that one actually matches up to the 10YR21, so that's an excellent color. If we would have colored that one across the way over there that's been managed differently, it probably was maybe a 3-1 for color, or a 3-2 or a 2-2. So excellent color in this particular soil. And if we start looking at soil structure, and I guess, Daryl, we have to go back and look at that surface again.
Okay.
And what we do is we look at the different soil structures that exist in the soil. So we mentioned before the granular structure, and that's what this would be. That would be granular structure, similar to what we find on the page here in this guide. If we start looking at some of that lower horizon, it probably doesn't really break into granular. It's more a subangular blocky structure. And then finally, when we were looking at some of the field, in the field next to us, we did see some of this platy structure, that horizontal platy structure. So those are the different types of structure that you might run across in some of these agricultural fields.
We are looking just upslope from where we were before. We're still in the 60-inch corn spacing part of this field, but what we want to take a look at here is you notice that the corn is a lot shorter here. I mean, it's stunted here versus where we were before. Part of it is, yes, it's very dry. We are in a drought. But the other thing too, the point that I want to make here is that the soil type that we are on here, it's a sandier soil than where we were before. So because we have the sandier soil, the soil itself cannot hold as much water as the silt loams, the silted clay loams. So because of that, we are seeing the impact of the drought on the crops. And you can see in the field kind of where this sandy soil resides on the landscape here. Off in the distance, you can see where the crop is taller, and it just sort of kind of goes around here, and then not too far away, we get into that different soil type. So when you're looking at soils, it's important that you know what soil you are on, what the soil texture is, whether you are in a sandier soil, clay soil, anything in between. So that's an important thing to know when you're going out and you're looking at these soil health systems. So what soil are you on? There are tools out there that we use. One we should be using is Web Soil Survey. On there you can pull up your map and you'll know what soil type you are on. You can go in there and you can also look at what the texture might be, as well as when you go out to the field, you can check it. So there are other tools we have on your cell phone. You can actually there's a Web Soil Survey app on the phone, so I can dial that up and I can get what soil I'm on, where I'm at in the field. So there are some tools out there, but you definitely want to make sure that you know what soil type you're on when you're doing these soil health evaluations, and then also just take a look what the impact on the crop.
Hi, I'm Jay Fear and Daryl Oswald. Daryl's the manager of the Menoken farm. And today we're just going to walk through a slake test, and it's something you can use for soil health assessment on a field. And it's something you want to start with a PED, and so just a clod off the surface. And Daryl's got one, I've got one. We keep a little tray of them here so that we understand where they came from. And really it's a collection of small soil aggregates glued together. And so the glues are made by the soil biology and the plant together. And if we have adequate glues, we have an integrity of the soil aggregates. So we would have soil aggregate stability. And to test this, you need some wire, a little netting. In this case, we cut the bottom out because we like to put a lot of pressure on the PED itself. And then a tall cylinder, something that holds water and is clear. And where the term slake comes from is when we put this PED in the water is going to come into the pore spaces and it's going to push the air out, push it out, should bubble up to the top, and it created pressure. And if the pressure is greater than the glues, it's going to start to peel off or slake off as the individual soil aggregates. So
It's important, Jay, to make sure your beds are dry.
yep.
And so obviously these beds are good and dry. And so as we move forward with the experiment, again, we're going to put the pad on the screen and we'll put them in and see what happens.
So I'm starting to see some bubbles come to the surface, Daryl.
Right, we're displacing the air, which is in the pore spaces in the soil, and so that's coming to the surface.
So now there's a pressure being created inside,
Right.
and starting to push against these soil aggregates.
And since I was the bad guy today, I get to be the bad soil, and if we look closely, we can see what's starting to happen. And
So a little history on these. The soil that I'm at right here, it's a long-term no-till field with cover crops and with livestock integration. And Daryl, your field?
my field obviously has been heavily tilled, no livestock integration, and no cover crops, and not much diversity. And so we're starting to see the results of our management.
So the cover crops make a huge difference in themselves because you, in addition to the CO2 taken in by your cash crop, afterwards you come in with a cover crop and you have a new start of CO2 that you take into the plant, and the plant ultimately putting the carbon exudates into the soil. And so then when we have the carbon in the soil, we have the food, and the biology can do its task. If we were standing outside right now and it was raining, we can get a huge amount of soil loss. But this is an inexpensive means of assessing your field. But the main thing is get them into a container and as Daryl said, dry them. So let them dry, get them into the sunshine a bit, and after they've dried a few days, then you can go ahead and start your assessment. And it's very, very effective at explaining what is and isn't happening in your field.
I'm Dave Archer. I'm the research leader at the Northern Great Plains Research Lab at USDA ARS. And I'll introduce Mark here.
Yeah, I'm Mark Liebig. I'm a research soil scientist also here at the Northern Great Plains Research Laboratory.
Yeah, so we're here at the Area 4 research farm. And this farm was established in 1983 to look at field scale research and farmer-centered research. And one of the things that we've had in place at this farm for a long time is a set of long-term rotations. And so I'll turn it over to Mark to talk a little bit about those rotations.
Right, so beginning in 1984, the previous researchers here established a springweed fallow and also some continuously crop treatments, and over the years we've added additional rotation treatments to really explore the effects of crop diversity on not only crop productivity and stability, but also a suite of different soil properties and so forth. And so it's been, the fact that we've been able to run this for nearly 40 years allows us to develop a data set that's really valuable and helps inform producers what the effect of crop diversity has on their bottom line.
So some of the things that we've looked at, we've had these long-term rotations from the crop fallow that Mark mentioned, all the way through a 3-year rotation, a 5-year rotation, and then a dynamic system where we use a wide diversity of crops, but we make the management decisions each year, the cropping decisions each year on that, based on the weather conditions, based on markets, based on previous crops, and all those considerations. If a farmer's looking at their crop yields on their own place, and they may be seeing their yields go up year after year, they may think that everything's going fine, but the reality is that maybe things are degraded and their yields could have gone up at a faster rate if they had done some things to protect their soils a little bit more. So that's one of the ideas, I guess, behind looking at these systems in the long term. We found that the more diverse rotations tend to be more profitable. They tend to be less risky and less economic risk over time. The other thing that we've noticed is that we're starting to see some trends in the different rotations where they're increasing net returns over time, and they appear to be increasing at a faster rate with a more diverse rotation than with a less diverse rotation. And so we think, a lot of that's due to changes in crop varieties and things, but we think a part of that is due to the changes in the soils.
Right, exactly. So, the fact that this has been long-term allows us then to look at change over time in the soils, which in a semi-arid environment like this, soil properties often change pretty slowly. And so, under those different treatment scenarios, we've been able to see actually an improvement in key soil properties, the organic carbon, the nitrogen, infiltration rate, where we have more diversity. And so that's important information to go along with the improvements in net returns, and maybe, as Dave mentioned, it may be explaining why we're having greater net returns, because those types of practices are actually improving soil condition.
In recent years, we've been including perennials in that rotation. So we're sitting in the alfalfa field right now, and we've included that as part of our long-term rotation. So we'll put alfalfa in, leave it in for 5 years, and then move it to another field. And we think that's long enough to provide some soil benefits, but a question that people often have is how these do economically, these perennial crops. And those have been very profitable for us as a part of those dynamic rotations. And so, we analyzed it with and without the perennial phase, and it was just as profitable with. And in many years, the alfalfa hay has been one of our more profitable crops.
Right. We've also done some research on cover crops and how they sort of fit in within a crop rotation, and some of the work that we've done recently is looking at interseeding cover crops in a standing corn crop, which in this dry environment like this, it might be considered to be fairly risky. And so we've done multiple years of those evaluations, seeding into standing corn to see what the trade-offs there with respect to any yield, any negative yield impacts, which we did not observe, but also on the subsequent productivity of the biomass from the cover crop itself, because you can almost think about that as perhaps serving a role of not only being able to capture excess nutrients, but also it could be a forage source for those producers who have livestock.
This is a place where we can try things in the long term and track it through and see what are the long-term effects of trying some of those practices.
Okay, well, now we're joined with John Hendrickson. John is a research rangeland management specialist here at the Northern Great Plains Research Laboratory, and he's done a lot of work in understanding the importance of perennials within cropping systems. And there's one project in particular we've worked on together, looking at the productivity and also some of the soil attributes. And John does the above ground work, I do the below ground. So.
Yeah, well, thanks, Mark, and I really enjoyed that project while we were doing it. It looked at basically 5 different perennial treatments. One was alfalfa, one was intermediate wheatgrass, switchgrass was one, and then there was a mixture of alfalfa and intermediate wheatgrass and alfalfa and switchgrass. And so the interesting thing I always felt with it was taking them out of production. We split that into 4 basic strips across there, if you remember, and took 1/4 of them out each time. So we had an idea on how long they had to be in production and also how long the benefits lasted afterwards.
Right, and it was spring wheat, right, that we used as the test crop, if I recall. And so over time we were able to see how the length of the perennial phase impacted, I guess, not only the amount of yield, quantity of yield, but also some quality aspects as well.
Yeah, that's correct. If we kept especially alfalfa in for 2 years, and broke it out, we got the same yield as we did with fertilized spring wheat. The longer we kept in, like alfalfa or let's just say anything that had alfalfa in it, the alfalfa or especially the alfalfa intermediate mixture, the longer we kept it in, the longer those benefits lasted. And the interesting thing with alfalfa is that it increased the protein in the spring wheat grain afterwards. And, you know, so, and at the end of the time, the quality of the spring wheat we had was, following a perennial, was the same as the quality of a fertilized spring wheat.
One of the other things that we worked on was on the soil, sort of pretty much near surface soil changes and so forth. And I think one big takeaway from that study that I recall was that where we had the alfalfa and the intermediate wheatgrass together, we were often seeing the sort of the biggest increase in labile or organic matter, the particulate organic matter, carbon and nitrogen. And we also saw some improvements with water stable aggregation as well. So we were not only improving some of that labile organic matter, but also seeing some structural benefits from a perennial inclusion. You spend some time putting a perennial in and investing in that, but then what you get from it is you get improved yield stability. You've got lower inputs, you know, after you've taken it out, and you've also done some things that are, you know, seem to be improving the soil as well.
Hi everybody, I'm Mary Podol. I get to serve as the State Conservationist for USDA NRCS here in North Dakota. And beside me, I have Mark Liebig, who represents ARS, or the Agriculture Research Station. And one of the great things is within USDA is our great partnership. And we're going to talk a little bit right now about soils.
We're doing our best with this soil archive here. There's a lot of it, you know, it's a great repository of a lot of history, and I think we ought to go take a look. We're here in what we call Building 24, more informally referred to as the Soil Archive Building. It's a place where we have over 5,000 samples, soil samples, that some of them go back as early as 1917, not only collected from here at the Mandan site, but from all over the Great Plains. And so, I'll show you some of what we got here. It's a real exciting place as a soil scientist to be, because you know, it's, you know, you can't get back time, and having these soil samples kept for us to be able to understand really long-term changes in soil properties is often a rarity. And so we consider this as a really critical piece of our research, essentially almost like research infrastructure, to look at the decadal impacts of some of our management practices. We have one of the oldest range management studies really in the world. It was started in 1916. And we have soil samples from that project going back to 1959, down to a 2-foot depth across all of the treatments that are still currently being practiced out on our south station. And as we go around the corner here, we come upon what we call the Haas Soil Archive. Howard Haas was a soil scientist here from about the mid-1940s to, I believe, 1974. He conducted a study looking at changes in primarily carbon and nitrogen across the Great Plains, looking at the changes in those attributes following the conversion of grassland to cropland. And so he worked with others, about 17 locations up and down the Great Plains, from Montana all the way down to Texas, collected over 3,000 samples. And we have all of them right here. It's an incredible resource. And so he did his work, great work, published in 1957, but we're finding that actually the archive we're getting a new life to it, because we've gone back to some of these sites in the Great Plains to look at changes now be over 7 decades, and that's a project that's ongoing. But again, you know, had we not had these samples, had he not insisted that they be kept, we wouldn't be able to ask those questions. You know, our methods that we use to understand soils, you know, 7 decades ago are not what we would necessarily use today. And so as our methodology, our instrumentation changes, evolves over time, we can use these resources to cross-check and to see how things compare. And so, you know, with that confidence, then we think that we can we can, I think, shine, a new light on soil change, long-term soil change across the Great Plains.
Well, that was an excellent day, wasn't it?
Definitely was.
The highlight for me was going to Menoken Farm and looking at the soil aggregation and the impact of the cropping system on the soil.
And for me, going to the Menoken Farm and looking at the high tunnel and what practices that they're using within the high tunnel.
I have to say, seeing that soil archive that they have, 1917, that was definitely a highlight for me.
For more information on soil health, visit our website.
And thank you so much for joining us today.